All right then, chapter 6 deals with cells, the parts of the cell, organelles, membranes, cytoskeleton, stuff like that. Maybe not the most exciting, maybe a tiny little bit boring chapter, but we're going to power through it anyway. All right, let's get started by taking a look at a little bit of scale, where we take a look at the sizes of some of these things that we're going to be talking about. So some of the largest cells are actually visible to the naked eye. You can see them like eggs from frogs and birds and things like that. We know that there's protista like amoebas, where a single cell is actually visible to the eye. Some of the larger ones you can see. They look like a tiny little dot made with a pencil, a sharpened pencil, about that big. But a lot of cells, the cells in our body, most of them require a microscope to see. They're very, very small, so we say they're microscopic. And many of them we can see with a light microscope, which is the most common kind of microscope that would be used in a school setting. A more powerful type of microscope then would be the electron microscope that allows you to see smaller items like organelles and maybe all the way down to viruses and maybe even larger molecules. All right, so the types of microscopes that scientists use, basically there's two main kinds, even though there's three listed here. There's light microscopes and electron microscopes. The stereoscope is really a type of light microscope that gives you an image in 3D. And it's not very powerful, so a stereoscope is limited to maybe about 50x, maybe 100x at the most. So you'd be looking at fairly large objects. The light microscope has a limitation of maybe 1,000, possibly, if you're stretching it, up to 2,000 times magnification. So the limit with the light microscope is not necessarily the lenses. We can build a microscope with lenses that are very powerful to magnify more than this limit that I wrote here. But what we're lacking then is resolution. And I'll show you what resolution is in a minute. That's the limiting factor, and it has to do with the wavelength of light. The wavelength of light has a finite size, and if you're trying to look at objects that are close to the wavelength of light or smaller, you're not going to be able to use light to visualize those. That's where the electron microscope comes in. Electrons have a much smaller wavelength, so you can look at smaller objects. There's two main kinds: the scanning and the transmission electron microscope. So the scanning electron microscope is the less powerful one, but it gives you images in 3D. It goes up to maybe 500,000 times, maybe even over 1 million now with the newer technology magnification, but it scans on the surface of the object and gives you a 3D appearance for the image, which is kind of nice and kind of neat looking. The transmission electron microscope is exactly what it says it is. The electrons have to pass through the object, so you have to have a thin slice of the specimen that you're going to study, and it only gives you a 2D image, but your magnification is something like 50 million. All right, so 50 million times magnification, which is very, very impressive. And with modern electron microscope, we can see large molecules, actually. We can visualize the molecule, which is incredible. All right, so here's some examples of, on the left, we see scanning electron micrograph, where we see the three-dimensional surface of some pollen grains. This would be a transmission over here, where we're looking at a thin slice, and we just see a two-dimensional image. I mentioned resolution, and a good example of a problem with resolution, like you can magnify things as much as you want, but it really doesn't do you any good. For example, if I take a thumbnail of my face, which looks pretty good if I do say so, and then take that exact picture and magnify it without making any other changes, this is what it looks like, and it just looks like a bunch of squares, just a bunch of dots essentially. And it's hard to tell what we're even looking at. So yes, this has high magnification, but it has low resolution, which makes it almost useless to magnify it that much, because it really doesn't add anything to the precision of the image. This is one of the gripes I have with TV shows and movies when they always have like surveillance footage of the bad guy in a crime show, let's say, and then they tell the tech in the lab like, oh, can you zoom in on that? Can you sharpen that? And then they magnify an image like this, and then they magically sharpen it so it looks crystal clear, which is, of course, fake, because that doesn't really work in real life, unless you had a 100 million megapixel camera. Anyway, all right, so that's the deal with the resolution. Electron microscopes give you magnification and resolution, and good quality light microscopes up to that maybe 1,000 limit, or maybe a little above that, if they have good lenses and good lighting, also provide magnification and decent resolution. All right, let's change topics a little bit. Let's talk about why we even have cells. Like, why are living organisms even made out of cells? What's the point of all these tiny little cells when we could have just maybe, parts in our bodies that are much larger units instead of these tiny little microscopic things where we have trillions of them that make us up? And the answer comes down to surface area to volume ratio. It has to do with transporting nutrients and waste in and out of the cell and the efficiency of that process. If you have a tiny little cell like this, it's real easy to get things in and it's real easy to get things out, for waste to come out and for nutrients like glucose, amino acids, things like that to go in and to diffuse to all parts of the cell to basically cover all the regions that it needs to cover. But if you had a really large cell, if that amino acid or that glucose or whatever it might be entered, it would take really long for it to diffuse to the center. It would maybe never even get there, which means the parts here would never actually get the required substances. So at the surface is where things enter and where things leave. So the surface is very, very important for this exchange. And large cells simply have a very small surface area to volume ratio. So their volume just gets too big for the surface to keep up. And this sort of fake cell diagram shows that really well. So we have a cube that's 5, let's just say centimeters, whatever the unit is, doesn't matter. It's 5 centimeters in, on edge, so it's 5 by 5 by 5. And here we have a cube that's 1 centimeter on end, so 1 by 1 by 1. So the surface area of the small cube is going to be the 6 sides to it's like a die, like when you're rolling dice. So we know the 6 sides. And each side, the surface area is calculated by length times width. So it's 1 times 1 times 6. This is how you'd figure it out. 1 times 1 is one side, times 6 is all the sides, gives you 6 square centimeters, let's say. I don't know if we need the units, but whatever, it doesn't really matter. Here we have a cube that's 5 by 5 by 5, so it's 5 by 5, which is 25, times 6, which is 150. Okay, so the surface area, you might say, well, it is larger than this one, so it has a bigger surface area. Well, sure it does, but the volume went up more than that, because the volume went up 5 by 5 by 5, length times width times height. So the volume went up to 125 cubic centimeters. Okay, over here with the small one, the volume went 1 by 1 by 1 centimeter, which is 1 cubic centimeter. So surface area to volume ratio is 6 divided by 1 equals 6. All right, over here we have 150 divided by 125, because we have that larger volume now, is only 1.2. So that's a much smaller surface area to volume ratio. We have way less surface area. If we were going to take this fake giant organism, giant celled organism, whatever you want to call it, and divide it up into lots of small cells, so we'd make, actually, how many would that be? 5 times 5 times 5, I guess 125 small cells. We would then figure out the surface area of those guys would be 750, because there's 125 times 6. Each of them have 6 cubic or square centimeter surface area. The volume would still be 125, because that's not any different. Whether you do this in one piece or in many small pieces, the volume is the same, so that's still 125. And our surface area to volume ratio is 6, which is the same as it is here. So by having lots and lots of small cells, we eliminate that problem of surface area to volume ratio, and we allow each cell to get the nutrients that it needs and to be able to diffuse them to the middle of the cell and to be able to eliminate waste. Because each of these tiny little cells would have a blood vessel, a capillary next to it, to supply glucose and amino acids and nutrients, and to take away waste. So here's a little exercise, a little math problem for you to do on your own. So we're going to do just a 10 by 10 by 10 instead of a 5 by 5 by 5. I'll let you do that math problem on your own and figure that stuff out. Oh, look at that. There's the solution. Well, don't look at that until you try it on your own first. All right, let's actually dive headfirst into eukaryotic cells. So we probably should define the word eukaryotic. It just means cells that have a nucleus and membrane-bound organelles, as opposed to, let's say, prokaryotic cells, which do not have a nucleus or membrane-bound organelles, such as bacteria. So let's start with animal cells, which is what's pictured here. This is sort of a cartoon example of an animal cell with all the parts. It's not really meant to be super realistic, like the nucleus may not be this big. This kind of cell probably wouldn't have a flagellum, which is shown over here. This is just basically they took a cell here and they gave it everything they possibly could give it, just to show everything, where all of these parts might not necessarily be in every single kind of cell. All right. Anyway, I'm going to go through each of these individually on separate slides, but this kind of gives you an idea. Hopefully many of these are familiar to you from freshman bio. All right, here's a plant cell, and my pet peeve comes true again, where any kind of textbook artist always draws the vacuole too small in a plant cell, but you notice that the plant cell has a cell wall in addition to the cell membrane, still has most of the other parts. It has chloroplasts, which are new, and it has this vacuole, the central vacuole, which is not in animal cells. In most plant cells, though, the central vacuole is a lot bigger, so this should really be drawn bigger, but then they wouldn't have space for all the other parts, and we wouldn't be able to see all this other stuff. There's also some new stuff here that we have never talked about before, like plasmodesmata. We'll cover those later. All right, so here's then an electron micrograph of a plant cell, and you might say, hey, wait a minute, look, the vacuole really isn't that big. You know, this proves that you were wrong, Volk, and the vacuole is actually much smaller than you were claiming, because when I draw a plant cell, I always do this. Why am I drawing a plant cell red? The vacuole is like this big. Takes up like most of the space in here, like the nucleus is like here and all the parts. That's what I'm claiming, and then this electron micrograph seems to prove me wrong, because it's like, what the heck, this vacuole is much smaller than Volk's claiming. Well, the answer to that claim is, my rebuttal would be, is what they did here is to take this picture. First of all, this is not a scanning, this is a transmission electron micrograph. This is transmission, TEM, okay. And what they did is they took a slice like this. They took a cut like that. So you just see a little part of the vacuole here, looking at the corner of the cell like this, and you're only seeing this little part of the vacuole here, when in the main part of the cell it's much bigger. See, I'm still right. Anyway, all right, so cell wall, chloroplast, vacuole, those are the main differences, animal cells versus plant cells. Shape and other stuff like that too, but those are the main ones. All right, why don't we dive into the organelles? And the first organelle we're going to talk about is the nucleus. And everybody kind of knows what the nucleus is all about. The nucleus has this double membrane. It has two membranes surrounding it. It's got DNA in there. The DNA here is called chromatin because the DNA is uncoiled. It's all sort of spread out over the matrix of the nucleus. So chromatin is basically spread out DNA and proteins, histones, and DNA. Histones are the proteins that the DNA wraps around when the chromosomes coil up. There's a nucleolus in the middle, and you can also pronounce that nucleolus. The nucleolus produces ribosomes for the cell that then get exported out to the rough ER and out to the cytoplasm. All right, so that's the function of the nucleolus. It's like a mini nucleus inside of the nucleus. The membrane, or the double membrane, the nuclear envelope we call it because it's a double membrane, actually has these little holes that are pores. They're called pores, and they allow ribosomes to come out. They also allow mRNA to come out. So messenger RNA will leave here after it's been made from the DNA in the process called transcription, and it has to exit the nucleus to get to the cytoplasm so that protein can be made. We'll talk about that in much more detail later. There's micrographs here of of the pores, of the membrane, all of that stuff. There's inside of the nuclear envelope, there's this thing called the nuclear lamina that's made out of intermediate filaments. And that just strengthens it and keeps it three-dimensional so it doesn't collapse. These kind of strong fibers made out of keratin. We'll talk about those later. Don't worry about that now. All right. Okay, the other thing that's shown here is the rough ER, which kind of connects to the nucleus, and we'll talk about why that's significant in a minute. So the rough ER is this folded, like baggy membranes that's all over the place here, and it has tiny little dots on it, which are the ribosomes. So let's discuss what the rough ER does. The rough ER is primarily a place where proteins are going to get made. Okay, so it's for protein synthesis. Now, the rough ER itself doesn't actually make the proteins. It's the ribosomes that make the proteins, the ones that are stuck to the rough ER. And the fact that ribosomes are stuck to the rough ER is what makes it rough. That's why we call it rough, because it has these little dots on it. It has a rough surface because of that. Ribosomes are technically actually two subunits. There's a small subunit, a large subunit, and they're floating around in the cytoplasm apart. They're not actually assembled. And once the mRNA comes out, the mRNA will bind to the small subunit, and then the large subunit also binds, and it forms this complex. So they clamp the mRNA in the middle, and then they read the message on the mRNA to make protein. All right, now the proteins that are made by the ribosomes and the rough ER, they get exported. Therefore, export. They get shipped out of the cell, or they go to the membrane, to the plasma membrane, the cell membrane on the outside. They stick in the membrane. It's either one of those two, either exported, shipped out, like for example, cells in the pancreas would make insulin, which then doesn't stay in the pancreas, but it gets dumped into the bloodstream. Though that insulin is made on rough ER ribosomes. All right, what's another function of the rough ER? Let's see if I have some place to write here. Yeah, I guess I'll just put it here. All right, we'll put it over here on the left, rough ER. I'm so well prepped here for this. Should have put a blank slide in. All right, so we already said like proteins for export. Another function is synthesizing membranes. It is the primary membrane factory for the cell. So most of the membranes in the cell were made by the rough ER, almost all of them. We call that the endomembrane system. So those are the two main functions of the rough ER. They have some other functions too, like lipid synthesis, like phospholipids and things like that. Makes sense with the membranes and storage, but these two are the primary functions of the rough ER. So membrane factory, membrane synthesis. And you can see that the nuclear envelope is connected to the rough ER here. It's actually literally like continuous over here. And that's because the rough ER synthesizes membranes that then get, fed into the nuclear envelope to maintain it and to, if it needs to get bigger or whatever after mitosis, the rough ER takes care of that. All right, the smooth ER then the smooth ER will actually connect to the rough ER like this, and we have that over here where there's a direct connection basically between the two because the rough ER produces the smooth ER. It makes the smooth ER. And the only difference is, well, okay, there's a difference in shape, but the main difference is that the smooth ER does not have ribosomes on it. So it's smooth in the surface because there's no bumpy ribosomes. And the smooth ER functions are going to be, the number one, most important one, in my opinion, is detoxification. Detoxification of poisons and toxic substances. That's really, really important. So the liver is just full of smooth ER. Liver cells have lots of smooth ER because they break down toxic materials for the body. So that's real important. Another function of the smooth ER is lipid synthesis, like steroids and things like that. That's similar to the rough ER also. Steroids and lipids like that are produced there. Another one is gluconeogenesis. So gluconeogenesis, as the word implies, so gluco refers to glucose. So that's kind of easy, glucose. Neo is new, like Neo from the Matrix. Okay, so Neo is new, and Genesis is creation. So it's glucose, new creation is basically what it means, and that's when organisms require glucose for certain body parts. Like humans, for example, the brain can only use glucose to function. So when there's not enough glucose available for brain cells, the smooth ER in cells will actually produce glucose from other molecules like amino acids and lipids. It'll convert them into glucose so that the brain has glucose to use, and the smooth ER takes care of that, which is kind of cool. How does that? And then finally, the smooth ER can be used for storage, like, for example, storing calcium, which is something we're going to bring up again in chapter 11. All right, smooth ER and rough ER. Those guys are kind of interesting, I think. I said this is going to be boring, but they're sort of interesting. I'm sure you agree. Kind of like these two. Golgi, okay. I guess it's all right. The Golgi apparatus basically takes the vesicles that'll come out of the rough ER with the proteins that were made, modify those proteins, and then ship them out to the membrane. Okay, so if the cell membrane is over here, these vesicles pass through the Golgi, the proteins get modified, they get packaged up, nicely, and then they fuse with the membrane out here. The Golgi has a cis side and a trans side. That has nothing to do with cis and trans fats. It's just happens to be the same wording that gets used. I think that comes from the Greek, but the cis side is the receiving end of it, and then the trans side is the opposite end. That's the shipping end. Okay, that's where the vesicles will exit the Golgi. And the Golgi is made by the rough ER because the rough ER is responsible for making just about all of the membrane organelles in the cell. So it's part of that endomembrane system, which is all based on the rough ER being the source of all these membranes. That's all I'm going to say about the Golgi. It's just too boring. All right, lysosomes. These guys are exciting because they're dangerous. Lysosomes are little vesicles that are also made by the rough ER, but these are special ones that have digestive enzymes in them. These are enzymes that can break down other molecules, which is kind of cool. So they're used to break down things in cells like a phagocyte, like a white blood cell or an amoeba, for example, would do this. And a food vacuole would come in. So food gets, taken in by the amoeba, let's say, or by the white blood cell, whatever it might be. And then the lysosome will fuse with that membrane and dump the digestive enzymes in there, which then breaks down the food. The neat thing about the lysosome is that the pH inside of the lysosome is really low. It's got a low, why am I drawing on the cell? Okay, I don't know. It has a low pH, and the enzymes work best. Their optimal pH is like, I don't know, low, like 2 or 3 or something like that. So what's good about that is that just in case one of these lysosomes breaks open, if one of them like pops open in the cell and it spills its enzymes out, what would happen is these enzymes would break down proteins and like important molecules in the cell. They might kill the cell. So they don't though, because the optimal pH of these enzymes is like 2, let's say. And the pH in the cell is 7. So if they spill into the cytoplasm of the cell, those enzymes will not work anymore because they're outside of their optimal pH, and they'll probably get denatured and stop working. So it's kind of like a safety mechanism that the enzymes of the lysosomes, the digestive enzymes, only work inside of the lysosome. Hey, that's cool. Pretty good. All right, so that's called phagocytosis, the thing I just described with the food vacuole and the lysosome doing that and all of that stuff. It's phagocytosis. So phago refers to eating. Cytosis is just cell. Cell eating is what it means. All right, and a phagocyte is a cell that will eat other things like a white blood cell, a macrophage, for example. All right, another really weird and cool thing that lysosomes do is something that's called autophagy. So auto is itself. Auto just means itself, and then phagy is eating. So it's eating itself. What it really is all about is that sometimes there's organelles like mitochondria, or here they show a peroxisome, that you don't even know what that is yet, but there's little organelles that are worn out. and they're old and maybe they're not working so well anymore. So what happens is a vesicle will form around those organelles. You'll form this membrane pouch around the organelle. The lysosome then fuses with it and breaks down these pieces that are in here, breaks down these decrepit organelles that don't work anymore. And then the molecules that are formed by that can be released and they can be reused to build new functional organelles. So that's kind of a nice little system that the cell has to take care of damaged structures. It can break them down and then use the parts to build new functional parts. We call that autophagy, self-eating, and the lysosomes will take care of that. Pretty cool. All right, here's the endomembrane system that I was referring to. It all starts with the rough ER. The rough ER is the source of all the membranes. It feeds membranes into the nucleus. It produces vesicles. It makes the Golgi apparatus. It makes the smooth ER. It even supplies membranes to the plasma membrane, to the cell membrane. Because these vesicles that fuse here will lend their, or not lend, but give their membranes to the plasma membrane. Okay, so a good portion of the cell membrane is made by the rough ER. Now, obviously, you have to have a cell to start with. You don't just start with rough ER, like forming a membrane from scratch. If there isn't one, you have to have a cell to start with, and the cells form through mitosis. But then that cell has to grow, and it has to grow into a bigger cell. And the way that works is the rough ER adds membrane to this small cell's membrane to make it bigger. So the rough ER has a hand in that and is obviously an important source of these phospholipids. All right. By the way, mitochondria and chloroplasts are not part of the endomembrane system. They have a different source. So not every single organelle in the cell is the endomembrane system, but most of the membrane-bound organelles are a part of it. Speaking of the powerhouse of the cell, the word mitochondria is plural. If you just have one, it's mitochondrion. Okay, so it's just one mitochondrion or many mitochondria. So if you want to say that slogan, you really should be saying the mitochondrion is the powerhouse of the cell, or mitochondria are the powerhouses of the cell. But that is a really simplistic, kind of a middle school approach to mitochondria. In this class, we're going to learn much more detail about how mitochondria produce energy for the cell in the form of ATP. We're going to go into excruciating detail. It's going to be awesome. Alrighty, so mitochondria have two membranes. They have that outer membrane, all right, and then they have the inner membrane. And the inner membrane has these folds, excuse me, these finger-like folds called cristae, which are shown here. The inside of the mitochondrion is the matrix. Oopsie daisy. That would be the matrix, which is the inner lumen or inner space of the mitochondrion. And then it has this intermembrane space right here in between the two membranes, and that's where a lot of, on this inner membrane and in the space, a lot of important stuff happens to make ATP. The reason we have all these cristae is to increase the surface area. We have a larger surface area in that inner membrane to allow us to make more ATP. Makes it really efficient. Also notice that mitochondria have their own ribosomes, and, which is not shown in this picture, mitochondria have their own DNA. They have a tiny little genome of DNA, like a loop of DNA, and they're very similar to bacteria. Interesting. Chloroplasts, those guys are found in plant cells. And by the way, don't be misled and think, oh, chloroplasts are in plant cells, mitochondria are in animal cells. Yeah, but mitochondria are also in plant cells. It's just that animal cells don't have chloroplasts. Anyway, let's see. Okay, chloroplasts have like a bunch of membranes also. They have this outer and inner, which really is not so interesting to me. What I really do care about is this way inner membrane structure, these little discs of membrane right here that we call thylakoids. These thylakoids is where photosynthesis takes place. It really doesn't want me to draw on here. So photosynthesis will take place there. PowerPoint is fighting me big time here. Oh, wow, stubborn. I'm even more stubborn though. Ha, dang it. Ha, got one. All right, so the thylakoids are all connected, and their membrane is where the light reactions of photosynthesis takes place. So that's the place where cells are going to take the energy from light. Okay, they're going to take CO2 and water, and they're going to be making glucose. Now, technically, the glucose is made in the stroma, but that's okay. That's fine. All right, so in between the thylakoids, there's the stroma, which is just the fluid inside of the chloroplast. A stack of thylakoids is called a granum, or plural is grana, not to be confused with grandma. And we'll talk a lot more about photosynthesis when the time comes. These guys also have their own DNA, and they are also not part of the endomembrane system, so they did not come from the rough ER. Peroxisomes are kind of an interesting, unique, tiny little organelle that is important for detoxification, specifically for detoxification of hydrogen peroxide. Hydrogen peroxide is something that forms when other substances are getting detoxified. What happens is when the smooth ER strips away like hydrogens from larger toxic molecules, accidentally hydrogen peroxide is formed because of all these extra hydrogens. So hydrogen peroxide itself is also toxic and can kill cells. So what has to happen is now all that hydrogen peroxide has to get broken down also. And peroxisomes take care of that. They break it down into H2O and oxygen gas, both of which are harmless, but even you could say beneficial. Both of those are nice. Hydrogen peroxide, yuck. Actually, we could do the Mr. Yuck guy. Okay? And the enzyme that takes care of that, we already know. Say it. Did you say catalase? You are correct. So this square is actually a three-dimensional crystal of catalase. There's so much catalase in this peroxisome, there's so much of it stored up in there that it started to crystallize because the concentration was so high. It went out of solution and started crystallizing. And that's why we see this square cube kind of a strange shape inside of the peroxisome, because that's where catalase is stored. And obviously certain cells have more of that because they are more responsible for breaking down toxins than some other cells. All right, that's a peroxisome. And we're going to take a break here and save this for the next video.